Hi everybody. Let's look at the material from chapter 22 in your text on macroevolution, or what is a species. Now, this might seem like a simple question, and a lot of biologists, or taxonomists in particular, tend to sort of reinvent artificial groups of organisms like class or order. We we know that taxonomic groupings change all the time, and so we have a sense that the taxonomy is sort of unnatural. It's invented by humans, but we've always sort of, or at least we want to believe, that species is sort of a natural taxonomic group. Species exist in nature. Classes don't exist in nature, but species exist in nature. So let's examine this question in a little more detail. Let's look at some pictures. I think it's pretty clear that we're looking at 4 different species of animals here, right? We have really significant morphological differences in these animals. At the same time, we know that all of the animals in this photo are most likely the same species. And this is because we're cueing into morphological differences. We're cueing into differences that we can see. But it gets kind of complicated. Here's 2 examples. When you look at these 2 different butterfly species on the top of the screen here, the monarch and the viceroy, they look very similar. These are ecological mimics, evolved this extremely similar morphology, and it's difficult to tell them apart. On the picture in the bottom, we see 2 birds, and they have some similarities. They have the same kind of body shape. They have the same beak color, but they're drastically different colors, their plumages. And this is not because they're different species, this is because of sexual dimorphism. This is a female, and this is the male cardinal. So whereas you would say the monarch and the viceroy are the same species, when they're not, you might say the cardinals were different species, and you'd be in error there. And this is because we have a sense of this idea of species as being individuals, or a group of individuals, that are reproductively compatible. More specifically, a group of organisms which can potentially interbreed and produce viable, fertile offspring. This species concept is known as the biological species concept, and it's going to be sort of our working definition of species. One of the things that I want to talk about here is some of the ambiguities in this concept. For example, here is a species of algae. This is called Euglena, and it is asexual. We see it in the process of binary fission here. It's just dividing. There's no sex going on. There's no reproductive compatibility to consider. So how do you distinguish one euglenid from another? The biological species concept doesn't work. Similarly, paleontologists cannot do mating experiments on extinct organisms. So if you are a paleontologist, you need another species concept as well. The biological species concept cannot be applied to all organisms. So we're going to spend some time talking about this in lecture. We'll discuss alternatives to the biological species concept. And we'll do this in a case study format, a very famous case actually, to illustrate the ambiguity of this term. We will also, and I'll introduce it here a little bit, but we'll discuss examples of particular types of reproductive isolation mechanisms. So we're going to fall back on the biological species concept, which requires reproductive isolation. And so we're going to need to talk about how reproductive isolation evolves. Anything that prevents mating or prevents fertilization will be a reproductive isolation mechanism. And even if that fails, even if fertilization occurs, there are also post-zygotic barriers. So let's look at these words real quick. Pre-zygotic means before the zygote. Okay, the zygote is the result of fertilization. Post-zygotic means after the zygote. Okay, so remember the definition of the biological species concept. It's a group of organisms that can potentially interbreed. Okay, so breeding is part of it, but it goes further than that. They have to produce viable, fertile offspring to be considered members of the same species. Okay, so let's go over these examples, and you can go to page 420 in your book, and you can look at this. One of the most straightforward types of reproductive isolation is habitat isolation, and this is illustrated by these garter snakes, which occur in the same geographic area, but one is aquatic and the other is terrestrial. If you're aquatic, you're never going to mate with somebody who's terrestrial. This is a habitat isolation mechanism. Temporal isolation is also a pre-zygotic barrier that prevents mating from occurring. Temporal isolation is isolation in time. And here we have these spotted skunks. So even though the ranges overlap, one of them mates in the winter and one of them mates in the late summer. So if you're mating at different times, you're never going to mate together. This is a pre-zygotic barrier. The last pre-zygotic barrier that prevents mating is called behavioral isolation, and this usually has to do with courtship behavior, which is very common in birds. The illustration here are these blue-footed boobies, which mate only after a courtship display that is very unique to their species. Okay, so if this courtship behavior isn't turning the female on, she's not going to mate with you. Now, these habitat isolation, temporal isolation, and behavioral isolation all prevent mating. If those fail, there's still two pre-zygotic barriers that prevent fertilization, and one of them is called mechanical isolation. Mechanical isolation is often incompatibility of genitals, or in the case of these snails, incompatibility of the genital openings. Okay, a second kind of post-mating but pre-fertilization barrier is gametic isolation. Gametic isolation is usually involved with some sort of incompatibility of gametes. For example, if the enzyme of a sperm cannot penetrate the egg, fertilization cannot occur. So here you have these sea urchins. The sea urchins release their sperm and eggs into the water, but it's difficult for gametes of different species, such as the red and the purple urchins shown here, to fuse because proteins on the surfaces of the eggs and sperm bind very poorly to each other. Okay, so these 5 habitat, temporal, behavioral, mechanical, and gametic isolation prevent fertilization. They are pre-zygotic barriers. If those fail, there are 3 post-zygotic barriers that can prevent either the viability or the fertility of the offspring. And the first one is illustrated here with these salamanders. This is called reduced hybrid viability. So the example here says that some salamander subspecies of this genus live in the same regions and habitat where they may occasionally interbreed. Most of the hybrids do not complete development, however, and those that do are frail. Okay, so this is reduced hybrid viability. This next one you're undoubtedly familiar with. It's the hybridization of the female horse and a donkey, which is called a mule. This is extremely viable, and in fact it's described in your book as robust, but it is infertile. It cannot produce its own offspring. In other words, the lineage stops there. So this is reduced hybrid fertility. The last post-zygotic barrier that I'd like to discuss is called hybrid breakdown. And this is when, like for example, here we see that some first generation hybrids are fertile, but when they mate with one another or with either parent species, offspring of the next generation are either inviable or sterile themselves. So these barriers prevent either mating, fertilization, or hybrid viability or fertility. So we're going to come back to this in class, and we're going to generate a list of some examples to sort of elaborate on this context, but I wanted to introduce it here. So we just learned that reproductive isolation is key in species formation or species divergence, and it's the very definition of what a biological species is. This can happen in two main ways. It can actually happen in a couple more ways, but I want to keep it simple here, so we're going to talk about two very, well, not necessarily common ways, but two ways that are highlighted in your book. And the first one is pretty easy to understand. Allopatric speciation is when species form while geographically isolated. So if you imagine we have this population in one lake that is sort of contiguous, and gene flow can happen between these two major pools here. If the water level was to go down, there was this physical barrier to gene flow, and you can imagine that maybe selection pressures would be different in this pond than in this pond, or random mutation would occur differently, and eventually some kind of reproductive isolation mechanism would evolve between these two species. So if this water level rose again and these species were allowed to interact with each other, they would not be able to produce viable fertile offspring. There would be either a prezygotic or a postzygotic mechanism in place that would prevent that. Sympatric speciation is speciation without geographic separation. And this is a little bit harder to understand. Your book talks about polyploidy in plants. I'm not going to dwell on that in class. We will instead talk about behavioral isolation and habitat isolation, and to a certain extent temporal isolation as being drivers of sympatric speciation. We'll examine this in the case study that we look at in class. So we will examine some of those things sort of via discussion and lecture. As I've mentioned, in lab we are going to examine behavioral isolation in two closely related bird species pairs: the eastern versus western meadowlark, and the blue-winged and the golden-winged warbler. If you look in a bird book, these are considered separate species. We will use their songs, which are used for, among other things, courtship, as a way to measure quantitative differences, and we'll do a little research on these birds to make predictions about which one should exhibit more differences in song. Okay, another possibility for lab this week is to look at dewlap coloration in Caribbean anoles. Male anoles use these sort of fancy dewlaps on their throats to attract mates.
And the coloration of the dewlaps can be used to distinguish males of one species between males of another. Now, the brightness of their dewlaps is dependent on the environment in which they live, and the strong selection for sort of divergence in male dewlap coloration can be a driver for speciation. So, depending on what I decide this semester, we may do the bird lab or we may do the anole lab. Both are interesting, so give both of them a thought.
So that's kind of what lies ahead. I hope you found this relatively straightforward. We're going to spend some time talking in class, but I wanted to get the basics out of the way before we did that. So I will see you in class, and thanks as always for your attention.